p.m. in the Netherlands. It's time to start. I have two very good news for you. The first one is Friday, and the second one, the last 2 hours before your weekend, are for talking about quantum for the medical industry. It is time to move on with our quantum industrial market pool. On 12th of March, we discussed quantum technologies making a difference in the future of medical solutions. We are now living the second wave of quantum technologies. On the first wave, it was all about quantum mechanics, and with that idea, semiconductor lasers conquered the world. So now we are on the second wave. We are talking quantum technologies having the ability to study every particle individually. At our last World Quantum Photonics Technology Summit in Barcelona, we could still travel, SAIS presented an internal study to argue why they would invest in quantum technologies. SAIS, as a market leader in microscopy, concluded that they needed to know how much information can be extracted from a single photon or a photon pair. So how precise and how sensitive could future sensors be if empowered by quantum technologies? So the purpose of the meeting on Friday, March 12th is to explore the answers to these questions. Do we need single photon sources, single photon detectors, and different optics? And what current and future off-the-shelf photonic components and systems are relevant for the future quantum medical equipment? Can we use quantum entanglement, the most important characteristic of quantum technologies, to develop microscopes with the highest resolution on the planet? And if so, by when? What is the role of Qnami, a brilliant quantum company, in quantum scanning magnetometry? And how would QSabre or Ranco also move this forward? And who do they need on board to realize their preferred future? And this is important. Can we effectively communicate this to leading enterprises like Abbott, Cambridge Consultants, or Merck? So let's unleash the power of quantum to explore the body with a single photon. Join our discussion with top specialists on Friday, March 15th at 3 p.m. Sign up early to participate in the Zoom room or watch live or later on the Epic YouTube channel. There are more than 300 hours of brilliant ideas in our YouTube channel. Check it out. All right, so it's time to start. It's time to start with our quantum technology meeting today. I have to say that I'm extremely happy with the feedback I'm getting from the quantum community for this meeting. Today is our third meeting, and these meetings are organized by the European Photonic Industry Consortium, the association to which I dedicate my life, and I dedicate all the passion that I have for the photonic industry. We are 15 people in the staff and almost 700 members. I can't wait to announce our member number 700, almost 700 members, corporate members supporting the photonic industry. I am speaking here on behalf of a team with marketing background, with technical background, 15 people fully dedicated to this industry. And what we do all together is to organize events, to provide access to a network, to help raise investment, to have the biggest website to find a job in photonics, jobsinphotonics.com, and to provide market reports to all our members. We are here for the sustainability of the European photonics industry. And today is our third quantum meeting. On the first one, we deal with quantum computing. On the second, we talk about quantum for security and defense, and it was spectacular, even though we couldn't show it in YouTube for obvious reasons. Today, we are back in YouTube to talk about medical, and the next one is going to be about transport and e-mobility. All those companies talking about single photon LiDAR, write that one down, but today it's about medical. And for those of you who know that we do more than quantum, write down the calendar for Monday, LiDAR applications. We have really a great agenda put together for the meeting on Monday on LiDAR technologies. Today at 3 p.m. we're going to talk about quantum for medical, and this meeting wouldn't be possible without the support of our dearest company in Finland. Our sponsor today is Modulight. Modulight, a company in Tampere, Finland, fabricating, manufacturing semiconductor lasers from the material growth all the way to the assembly and putting into products for clinicians. Thank you very much, Modulight, for sponsoring the meeting today. And the most important person in the room today It's not GE, it's not Merck. It's Dr. Sana Pica, who worked extremely hard on making this meeting possible. Sana, thank you very much. No, Elena Beletkaia, thank you very much, Elena, for working so hard for the medical industry. You are an expert in medical technology. Thank you very much for all the work you have done. What's in the menu?
I must say I'm very glad that you did mention Sana, because without her help, this meeting would not be possible. She is our quantum expert. So on the menu today, we have 6 excellent speakers covering different topics of quantum technologies and how those quantum technologies can help future healthcare. But what is even more interesting is the supply chain, all the companies and people that we are having in the room. And in the supply chain, we have people who are working with on quantum computing and quantum microscopy, and those who are manufacturing medical devices or other healthcare products who are interested in these technologies. Along the value chain, obviously, we also have representatives from photonic integrated circuits, photonic components, spectroscopy, lasers. We also are supported by a lot of R&Ds and system integrators, people who can put all of these components together to make them work. On top of that, people who have some market intelligence and provide consultancy, and a few pilot lines who are also helping to put new technologies into the production. So I'm very excited to see what new we can learn together, but even more how we can work together to make this happen.
Thank you very much, Dr. Elena Beletkaia, EPIC expert for medical technology, and Dr. Sana Pika, EPIC expert for quantum technologies, who've done a fantastic job. This slide only represents the companies who registered for the meeting today. So you're an EPIC member and you miss your logo on this slide. That only means that you forgot to register. Don't make this happen again. Always register for upcoming meetings. And today, because it's a quantum meeting, today we have something to try. Today, for the first time, when the meeting finishes, we're going to get a link in the Zoom room chat, which is going to give us direct access to wonder.me, fully for free, for us to mingle, for us to talk around, for us to network in a virtual environment. All you have to do will be to click on a link in the Zoom chat, close Zoom, and just follow very simple instructions on your browser, and then we'll meet each other again in a virtual environment for us to do business and to talk about our passion, which is quantum technologies. This meeting is, of course, also live streamed in YouTube. So hello, YouTubers of the world. If you want to get in touch with any of the participants today, send me an email, jose.pozo@epic-asoc.com. I will be more than happy to introduce you to any of the participants today. Also, if you have any questions during the meeting, post them in the chat in YouTube. I will read the questions in the Zoom room. And this is, of course, also valid for the people in the Zoom room here today with me. If you want to use the internal chat to talk to each other about potential opportunities, you will make me extremely happy. If after the meeting you didn't get a chance to talk to each other, send me an email and we'll make the introduction. I think that's all. Let's start with the real business. Today, we wanted to start the meeting with one company to raising a lot of interest for the quantum microscopy world. Today, we have, as an opening speaker, we have the honor of having Mathieu Munsch, CEO of the company Qnami. You already saw it in my introduction. We're extremely excited on how we can help one of the rising stars in the quantum microscopy world. The attention of everyone today goes to Mathieu.
Thank you. Thank you very much. Thank you very much for the invitation. Thank you very much for the enthusiasm. It's a pleasure to be here. I'm going to share my screen. It should show up in a second. Just let me know when this is through.
It's perfect.
Wonderful. So I thought today I'd like to introduce why quantum sensor technology will transform our world and how Qnami aims to support the development and become a leader in the quantum sensor market. So there's a wide range of industry at the moment undergoing rapid transformation. And just taking a few examples here, in research and semiconductors, new technologies are being explored to reduce the energy footprint, which leads to new needs in terms of measurement and inspection tools. In healthcare, I think the pandemic has highlighted even further the need for fast, precise, and early diagnostics. So methods to detect ultra-low concentration of molecules or improve medical imaging techniques are also needed. And with their boost in sensitivity and also their built-in accuracy, quantum sensors kind of open a window into what has never been measurable before. There's actually a range of different options for quantum sensors, and I'd like to focus today on the leading technology, which is using NV centers in diamond and the technology that Qnami develops. In this simplified roadmap that you have here on the left, I think the first thing that jumps off the page is that this quantum technology exists. It is already in the market, mostly for scientific usage right now, but already exists and is making a difference in the market. Generally speaking, the challenge in this industry is all around quantum sensor integration. So when we want to move from microscopy solution, which I'm going to talk a little more in a second, to more general purpose sensors that can be used for medical imaging or chemical detection, the ability to miniaturize the technology is important. To control the energy footprint and the power consumption is critical, which translate into questions around manufacturing cost and reliable high volume production. Now, the really great thing is that Europe is established as a leader in quantum sensor technology in particular, and I'd like to even stress out what I like to call the quantum diamond valley, which is this region in Germany and Switzerland going from Stuttgart to Basel, where there's really a historic pioneer development of that technology. So at Qnami, we contribute to core development of the technology in our quantum foundry, and we apply it to specific markets. We have a very unique team of soon 15 people gathering a wide range of expertise, going from material to software to instrumentation, and strong partnership, both with leading academic institutions, but also with industry to tackle both the scientific and the technical challenges that are ahead of us. And I'd like to conclude by giving two examples of products that we have been developed and connect with the topic of today. The first one is the Qnami Proteus Q, this first quantum microscope that Jose was referring to in his introduction. It's currently used by material scientists to image magnetic features on the nanoscale with applications in future computing, but potentially also in life science for the image of magnetic bacteria or even nanoscale NMR. The second application is biosensing chip, a new type of chip that we develop in our quantum foundry, which are designed for biosensing and applications in immunoassay detection or nanoscale chemical compound detection. This part is something that we do on a surface base, is tailored, it's flexible design so that we can adapt to the request in industry. We are trying to deliver that as a new business, separate business line in our company. So with this, I'll hand over and open. I'll be happy to answer any types of questions. And if you want more information on the products, you can just go on our website as well.
Thank you so very much for this great start with a great technology. And well, as it's an epic meeting, let's start an epic meeting also here with an epic question. And by your smile, I see that you already know it's coming. So the epic question asks what you can do for them and what they can do for you. We have a big supply chain of photonics companies here and other companies who are interested in collaborating with you as well.
There are many things to do. Let me give you one example. There is at the moment an application around brain interface, the ability to use quantum sensors to image magnetic signals in the brain. This is a very complex problem that takes the new technology that we bring, but also a fine understanding of the applications and the type of measurements that need to be done, what is clinically viable, interesting. So here we do need expertise from the very end application so that we can tailor our development in the right directions. So when I showed this kind of little house before with the quantum foundry and the different applications, our core know-how today is really in this base, and we try and partner with people in these different verticals when we reach out for new application.
Okay, we do have some companies who are working with medical devices, developing and integrating medical devices in the room. So as we already know, we have Sergey from GE Research. Sergey, would you want to share with us once again, so what GE is looking for in quantum realm, and how maybe Qnami sensors can be helpful, or what do you think about it?
Okay, so I was more planning to be in background and just listen to every presentation and look on the technology. So, and when, yes, from this presentation, it's certainly I did not understand what specific technology of Qnami is offering. So I have to spend some time just go in detailed and try to see how it will fit, what specific we can do with this technology. So without that, it's just, I cannot tell. So it's
okay. Sergey, one thing, because for me, when I look at this from the perspective of companies like Merck, like GE, I always wonder what do you see in this whole industry, whole quantum industry at large? Do you think that they are answering the right questions that GE and the medical companies are answering, or do you have doubts about the maturity or the time to market of these potential concepts?
So with all this medical specifically, so there is big road or long road between the real market and some technology. So obviously you have a few intermediate steps which you have to do. Okay, so first you have to demonstrate that this is specific sensor is measuring some specific characteristic of the body. So, and later you can just investigate how you can plug it in the real medical application, and after that build the prototype device, and after that only it's going on this real device. So like measuring magnetic field with very low levels, it's people already trying more than 10 years to use it for cardiac studies and for brain development studies, and there are prototype devices. And we know them, and the question, okay, if this, so you can approach this from two sides. This device is working, so technology is working, but it's very expensive because it was using squid technology, superconductive, all this very expensive. It's, I wouldn't say complicated, but it's very expensive. You have to really just do it very this specific way. So now you're approaching this problem and say, okay, I can do it for 100,000 or 1,000 times cheaper. Okay, and this I will solve the same problem. So this one approach. The second approach, you say, no, my specific technology is open totally new venue, and this opens like really big discussion how we can use it. So it's quantum technology is so broad, just title. So you can say, okay, where is quantum technology will be applicable next? Yes. So
we're going to try to answer how we can apply quantum technologies, but what I want to really hear from you, but also from Thomas from Merck on the performance of materials from Cambridge Consultants, is to really know how we can help these innovative concepts. Qnami is one of the most innovative concepts that we have in the European industry from the quantum microscopy. With the support of Horiba, they actually made something amazing and they put it in the market, which is huge. But I want this to be seen as an answer to questions. Mathieu, when you hear about Sergey's approach, like it's, okay, we need to find out, we have a problem to solve, we need to find out how long is it going to take you to solve it. What is the first thing that resonates in your mind?
It's quite interesting actually what Sergey just mentioned, and it probably highlights this need that we have. You mentioned magnetic sensors that can be used for cardiac or brain or imaging, and technology that exists. Indeed, it exists, and you were referring to this expensive squid technology. We have identified a range of properties that would benefit the field of, for example, brain imaging. For example, the fact that we can have a wearable system instead of something which is very rigid, and therefore just more simple to use. the question for us is that it still is a challenge to build such sensors, to ramp up the state of the technology to a say good prototype level. And for us, the question is to understand how valuable the difference we make is for the industry, and whether we can cross that gap. So it's a technical question I would have for Sergey, and probably the answer is not now for the forum. But I think this the connection between what we can potentially do, but the investment, we need to understand whether there is a value for the industry.
Let me go to one of the medical companies in the room, in this case, Performance Materials and Life Science Company, Merck. Thank you, Thomas. Thank you so much for being with us in the room. Merck GE, it's so great to have you here. Thomas, you're hearing that we have a quantum community. They are pushing a technology that is revolutionary. How do you see it from the eyes of Merck Performance Materials and Life Science?
Yeah, and healthcare. So, yeah, I'm a bit in the same situation as Sergey. In principle, I just wanted to listen in, and
never. This is an epic meeting.
I know, yeah, sure. And thanks, Elena, for making it happen. I think I opted out for being on a panel, but now let's be in the discussion. I think what is really cool is bring the worlds together. And Mathieu, really thanks for this great presentation. And we have also in healthcare industry, so for the healthcare pharma company industry association, we have recently found a network called QFarm. Yeah. And I'm not sure who else is from healthcare industry. I think I'm kind of alone. But we are looking at the problems of quantum, let's say, computing, but also at other technologies. And being a tech scout myself in our company, see it from my hoodie. We are also looking at sensing. Yeah, so I think this is really super interesting. It's tough to understand what the additional value is from this sensing technology. Same as Sergey said, and we have had a conference 2 years ago where we founded somebody from who's doing magnetoscopy. Yeah, and I think these NV centers are super interesting for doing magnetoscopy, for example, for biobanks. If we look at one of the future values of the healthcare industry, so my personal opinion, not speaking for the company, but is that we have biobanks, we have patient tissue, we have digital pathology. And with this digital pathology, we often just take it and freeze it and slice it with a cryo microscopy, and then they're gone. Yeah, and if we would have something that is reusable, eventually contactless, without the need to stain the material, this would be really, I think, interesting for applications to do more digital pathology, enable that we can reuse the tissue, etc. So this is definitely something I find interesting, and also the other topic you mentioned, the biosensing. Yeah, so I'm not sure yet how this would work, but definitely something for detecting, even in the lab environment, that we have some smart nose. I think everybody's talking about smart noses. People are now doing studies, even with quantum computers, who claim that they can smell COVID. Yeah, so a lot of topics are coming up, and I think if we would have a better, more intense dialogue with producers, sensors, material producers. On the one hand, what materials would you need, and on the other hand, how can we really make it live? Yeah. So how can you produce things that make sense for us to use? I think these discussions need to happen, but I doubt that this is the right time that we go in these deep discussions. But
of course not. This is to get to know each other. Mathieu, would you like to comment?
No, I think it just resonates well, and I would be happy to follow up a bit and try and explore how we can link cross the gap. Yeah.
One of the things that you're gonna like on the rest of the meeting is we're gonna hear concepts that in my mind are very close to the industry in terms of maturity, but very far from the industry in terms of market pool. And for me, one of the key companies that we have in the segment is Cambridge Consultants. I'm gonna keep coming with him after every presentation, but especially after the next one. We're gonna go now to the UK. We are going to go now to the University of Leeds, and we're going to call a person who I have a very, very high estimation for, a person I really look up to, Professor Jean Joseph. Thank you very much. Thank you very much for accepting this invitation. You have been for many years one of the key pullers on bringing quantum sensing on to the medical industry, a very difficult path, and today you're here to talk to us about how we can help each other. Professor Jean, thank you. The floor and the attention of everyone goes to the University of Leeds. We have you muted. I have actually a Mac to celebrate that. You are muted.
Yeah, got it. Yeah. Sorry.
It's coming. It's not yet here.
I think I shared the wrong screen.
Let's see. I think we're having some problems to get your screen on our screen.
It did work before.
In the
So
meantime,
it
I'd
will
like
work
to say.
again.
In the meantime, I'd like to say that Jean Jose has actually been doing many entrepreneurial efforts. Let's keep trying. I will just keep talking so that we don't get the silence in the room. Lots of entrepreneurial efforts to bring sensors in the medical segment for quantum, also for high resolution. Let's see what we can try again. Otherwise, we can share the screen from our side.
All done.
Yes, it's here. Let's go to slideshow mode. And it's perfect. The floor is yours.
Yeah. Thank you, guys. Thanks for waiting. Good afternoon, good morning, good evening, where you are. So I quickly go through these slides on the non-invasive sensing that we are trying in our research group, and it's recently being kind of being taken over by NICS. It's a company funded by one of our PhDs and being postdoc, and this Nick is. His name is actually Nick. The Nick is leading NICS. So in terms of sensing, this is the problem that we try to address, the sensing, non-invasively sensing glucose. So it is again using photons to sense glucose. The current technique is used to take blood out and measure it using a strip. So the issue here is to get the photons to interact with the glucose, blood glucose through the skin. So there are several methods people have tried. We tried one of these quantum principles into to address this particular issue of selectively detecting glucose at certain depths. So the idea is to find a wearable kind of device, develop it into a wearable kind of device, which can be used for continuous glucose monitoring for people with diabetes. So the principle of sensing is to try to detect a particular atom that we use as our probe atom, and then we want to detect a molecule in this case, which is the glucose molecule. So we need in our initial prototype, we tried to detect close to the skin or the surface of the skin, very close to the skin. So we need to get deeper into the skin. So what we used is some of the principles developed by one of my colleagues in the quantum optics group to extend this interaction into the skin. So that's basically using a simplified version. It's using a semi-transparent mirror. So what we are measuring is the photon decay from our sensor material. So the material essentially consists of a doped glass, which is a fluorescent ion doped glass. So this decay of the photons produced in this thin layer of fluorescent ions in the glass is what we are monitoring. And we have to extend the interaction of these ions or the photons generated by these ions to the deeper layers of skin and specifically to glucose. So a simplified diagram, this is how we initially tried the finger touch meter, which is a direct contact measurement. Now we modified this model further to enhance the fluorescence decay more sensitive to the glucose concentration, to make it more sensitive. So in our earlier studies itself, it was promising, but it was not sufficient to take it forward into a commercial prototype development. So we went back into the physics and refined the model. And we currently testing it in the lab to come up with the next generation of the prototype and to clinically test it. So there's some comparison of our early prototype, but the drawbacks were we need to fully establish the specificity of our sensor and also the calibration procedure for that. So we currently developing this in the laboratory stage. We went back to the lab again and we refined our sensing chip, and we are also requiring much more better detectors for accurately measuring the decay in the region that we are interested in. So our Nick is currently leading the business development and the product development research. So we currently testing it in the lab and hoping to have a next prototype in the nearer future. So that's the team we are working on. So Nick's, I said a few times his name. So Nick is here. He is a prosecuted fellow with the quantum technology supported. And also got other members of the team here. So Helmut is helping with the theoretical quantum optics side of things. And Ben is also working with me in the lab most of the time. So that's all about a quick run through the product that you are working on, which is enabled by the quantum technology. So thank you very much. Thank you, Epic, for providing me this opportunity. Thank you.
Thank you so very much for your great presentation and showing us some new technologies for such everyday use actually device as glucose measurement. So you mentioned in your presentation now that you are looking for new detectors, that you're in need of more sensitive detectors. And we have Richard here from Excelitas, a company well known for its detectors. Richard, would you like to comment?
Sure. I must say, I'm also getting used to Jose's methods of operation. When I see a chat message coming up from him during the end of a talk, I think, oh God, I'm up next. So it's always interesting to see. I mean, for us, yes, we've been making the single photon detectors for many years, and I'm here much to try and understand how we can take them to the next generation and what people need from a single photon detector. We know that we have a very good detector. We also know it has limitations. And I'm really here just trying to see what potential customers moving forwards are looking for. So I'm almost more asking questions than offering. But I mean, what can I say? We have, a silicon-based, so it's great for visible, but obviously somewhat large. And I see a nice image of a bracelet or a smartwatch. And I think, right, obviously downsizing is going to be key for any detector that's going into this. And the real question becomes how you can handle really low light levels with such a small detector and so little behind it. And that's obviously a huge challenge because, right now our detectors are about the size of my hand. So it's not really going to, lend itself to this type of application, but we need to know how we can move on.
Professor Jing, when I see this, I mean, first of all, congratulations, because it's truly fascinating. I am amazed. So, when I see this, I have the same thing as Richard. I just get goosebumps. So we have this glass that you impregnate with fluorescent ions, and you're actually we are actually exciting fluorescent on the finger underneath the tissue. How is the collection of those photons? Where is the detector? Where do you put the detectors, and what kind of detectors are you looking for?
Thank you. So basically the detectors go on the other side. And so that's why you got the glass and can get it to the other side. So both the pulse laser and the detector goes on the back side of it. So in terms of detection side, so we mostly looking at more accurate detection. I mean, it's not about more sensitivity probably. We have more photons, but it's about repeatably and more accurately measure it. And again, slightly longer decaying photons. So, let's say.
When I see the product, when I see the product, especially you compare it with the state of the art, and all this prick and invasive and sticking and all this horrible thing, and all you have to do really is press the finger against the surface. So why isn't the industry completely crazy about it? What do you need to tell the industry for them to say, okay, let's do this together? What do you think is missing?
I think getting the technology to the right level so that industry say, hey, we can take it now. So until then, in this sector, it's hard to, hard to get to the industry. So basically, we've been spending a lot of time in the lab to get it as good as we can in terms of detection ability. So because most industries or investors are not interested in doing the research phase. So it's
well, what these companies are very interested in getting a glucose monitoring sensor that you can just press the finger against it. Yaniv from Technion, I think he has a question or a comment.
Yes, I do. So I have a lot of questions regarding the technology. One is why use fluorescence and not a laser? The second is what is the mechanism in the glucose that is important? Are you shining in the resonance frequency of the glucose which absorbs the light? Or is the glucose a good scatterer of this specific frequency?
Yeah, in terms of the frequency, it's more to do with the resonance frequency. And in terms of your question about why don't use the direct laser, it's about how do we want to interact with the glucose molecule. So if you use the direct laser, it's more you getting into the spectroscopy mode, and which's been tried and got a lot of it's not as as good as we can to be commercially successful in terms of its performance. So we need something to basically address the molecule directly. So can we talk the molecule direct to the molecule from a distance? So you're creating a long range interaction in a sense. Basically, you want to that in the short range you can have the resonance interaction, but can be enhance that interaction to a longer range. So you are so specific to that particular molecule.
Okay, thank you.
Yaniv, keep them coming, because I think these are the kind of questions that we will want to have at this kind of meeting. So please do come in. Thank you so much for being here. I have to go now to my friend from Cambridge Consultants. I waited 37 minutes until coming to you. Edmund is our entry point for quantum technologies at Cambridge Consultants, which, as many of you know, is the key company for the TRL5 to TRL7 on medical device manufacturing that we have in Europe. Edmund, did you see the presentation from Jean Jose? And most important, how can Cambridge Consultants help bringing this concept to the medical industry?
I did see the presentation. No, thank you very much, Jose. Thank you very much, Jin. I think it's really interesting, the technology. The question is always in these sort of technologies, how do you take it from this lab-based demonstration through to a device? I mean, when you have this sitting on a patient's wrist, you need this to be taking robust glucose measurements over a long period of time and really trying to turn the technology into something which can be used in the real world. This is something that we do, but I think the first thing we need to understand in doing this is where is the technology now? What are the things that it's sensitive to? What are the developments that need to happen in order to make this robust? And that's really something where we can only really make that assessment with things in the lab. So I think in terms of getting to know the devices and the technologies that everyone here is going to present today, from a, in the lab-based setting is our first step to taking this through to a proper technology, or to a technology which is going to impact the lives of the patients with these diseases.
Gee, you know what to do now. We need to make an offline introduction because I need to make one of your prototypes in your lab. I need to put it in Cambridge. But I want to go now to Ulrich from SAIS. Ulrich, in the beginning of the meeting, I started in my introduction talking about your amazing presentation that you gave in Barcelona in our quantum meeting. And there you actually specified some questions that you wanted the quantum industry to answer in terms of can we get more information from future quantum microscopes. my question to you is very simple. How do you see the whole quantum community evolving towards answering the needs of the market leader in microscopy size?
Well, I guess it's a mixed message as always, right? So, so I just had a look before who's talking. So, if you look into NV center applications, so I mean, in still basic research, it's pretty straightforward. Yes, that's going to happen. Still not quite clear how big the market for that will be. So I think in that specific case, it's actually looking pretty good. Now, if you go beyond NV centers, like what we are interested in is, can we do better super resolution microscopy with quantum technologies? And there, I think one is working a little bit on the margins. So progress goes incremental, and it's and there quantum technology might not be in the center stage actually. So it can help, but just if you do a clever algorithm, just classical stuff, that might do the heavy lifting actually in many cases. So that's a little my mixed perspective on that.
I'm going to come to you after the next presentation. This was an introduction, because now we are going to go to Israel. We're going to go to Technion. We're going to go to Yaniv, which is from one of the strongest groups that we have in Europe on quantum microscopy. Thank you very much, Yaniv Kurman, from being here all the way from Technion. You are leading many of the fields on the quantum activities in Europe. We want to help, and we want to hear how we can help. So this is your task, to tell us how we can make you being even greater. The floor and attention of everyone goes to Israel.
Yes, so thank you for the introduction. I must say that I'm not the leading person in the research group. This is a group in Technion that is led by Professor Qnami. Now, in my talk, I'll present the ultrafast electron microscope, which is a very powerful experimental tool that we have in our lab. I'll explain a little bit about the basic concept and how this electron microscope is working, and I'll show the real front of the research and the capabilities of this electron microscope. So let me start by explaining the basic concepts of how it's working. This idea of having an ultrafast electron microscope was promoted also by Ahmed Zuhail, and also several other colleagues. He got the Nobel Prize at 1999. He will deserve it. The idea is to take an electron microscope, which has higher spatial resolution compared to optical microscope, and combine it with the femtosecond laser. So the idea is that you have a pump probe apparatus, so the laser can excite something in the sample, and then the electron can probe it in a high resolution. The way it looks in the lab is the following. So this is the same thing, just in the center, it's the illustration. On the right, it's how it looks in the lab. And in the center, you see here the column of the electron microscope. And all the energy comes from the femtosecond laser. The idea is that the laser goes around the optical table and enters inside the electron microscope and excites something, some sample that we have inside. Then the same laser is converted to UV and go excite a tip that releases an electron. Then the electron can sense whatever excitation was excited by the laser. Now, you see that we have here a change of the time delay, and therefore, once we change the time delay, we can understand the dynamics. So both the dynamics of some excitation in the sample and to understand it with very high resolution. So this is the basic concept. Now, what you can see with this experimental setup, one thing that you can see is to see, for example, movement of nanoparticles in a liquid. So you see here these rotations. Another option is to see mechanical movements. So for example, phonons, this is in a picosecond timescale, mechanical movements or vibrations of materials. Now, in these three examples, what we see is just the image of the electron. However, it could be something that is not that sensitive, that the electron is not that sensitive to. So something that is hidden inside the sample and it's hard to see. And for this, we need to have another energy filtering. So with the energy filtering, we can reach things that usually it's very hard. Let's say, for example, work that was published last year in Nature, that we managed to see how light behaves inside of a sample. So usually with all the electrons, without energy filtering, you can see just a sample of a photonic crystal. But once you do an energy filtering, you see the light modes inside. Now remember that this light is excited by external laser. Once we change the laser polarization, we excite different modes of the light in the sample, and we have different images by the electron microscope. Now, all of you, or most of you, might know that if you have a sample, the sample can have several different resonances in different frequencies. Now, usually you shine a laser, you change the frequency of the laser, and you understand what is the frequency of each resonance. But an ultra-fast electron microscope can actually image each one of the resonances. So you can excite a specific feature inside of the sample and look at it and actually not only have an image in one point in time, but see how it moves in time. And for this, I show here an example of a measurement of mine from previously this year, where we've measured how light propagates inside a 2D sample. So a sample which is atomically thin, but light can still move inside of it. And this is what you see here in the bottom right. Once we change the time delay, we excite the light mode on the edge of the sample. And once we change the time delay, we can see how the light is actually propagating inside of the sample. So we could see in live imaging how light is propagating. And generally, if I think of any sample, we can think of having an excitation of the laser, a single frequency, and look at a single feature of the sample and to see how it behaves and moves in time. Now, what is quantum in this measurement? If we look at this image of the electron energy after the interaction, you see here specific peaks. Now, the fact is that these peaks are divided from one another by a single quanta of photon energy. So we have a laser that actually changed the electron energy by specific peaks. Now, if you think about it, each measurement has a single electron. So we create, during the interaction, a frequency comb of the electron. And recently, last year, we managed to measure a free electron energy comb. So we have a sample, and we created that a single electron would have a superposition of energies. And this frequency energy comb could be very exciting for medical imaging, or actually looking at biological samples. So here are several examples of frequency combs, how they can help, for example, biological imaging or to trace molecules. In our case, however, it's a free electron energy comb, which we expect it to be much more sensitive. So we think that with this new finding that we have, we can actually help something in the biological industry. So with this, I want to finish. So I presented the ultra-fast electron microscope, the basic concept, and I've shown several capabilities of the electron microscope to create a free electron energy comb and to have a spatial temporal energy filter dynamics. And for example, here where we showed the evolution of the light itself inside the sample. Yeah, so with this, I want to finish. I'll be happy to answer questions.
Great, thank you very much for your great presentation of ultra-fast electron microscope. I have a few questions to you, but as usual, we'll start with the epic one. So what you can do for them and what they can do for you. So what cooperations you are actually also looking for, how other people can help you to make the system even better.
Yeah, so for me as a researcher, it's always a trade-off. You know, you want to improve your experimental setup, but you don't want to ruin what the specific capabilities that we have. I think that for us, when we do research, is to understand also what is needed by the industry, because we can have several different samples. For us, it was most of our research was fundamental physics. However, we can take it to different directions. And once we understand, maybe we have interesting samples, biological samples, and maybe, you know, if we have a drug or something inside that can help also the industry, and then to have a back and forth, I think it could be very interesting and exciting.
Okay, in that case, let's actually address to the industry. We have Thomas from Merck with us. Thomas, what do you think about this system? How do you see maybe it can help you, or do you have a question?
It can definitely help. Just the question is how. I think it's a fascinating presentation. I was just a bit lost on actually what the sample would be, because in my past when I did physics, the electron microscope was always dependent that we have this in vacuum, and you do some I don't know, coating with metals, etc. And I recently had the pleasure to listen to a talk from Stefan Hell, who is doing also nano resolution with fluorescence. So question: are you working with samples in wet? So could we apply it to single cells in a living system, or is this dead?
So nothing is alive in our samples. Mm-hmm. But if the sample itself could be encapsulated somehow, like closed in, and would not contaminate the vacuum, then it's definitely possible. Because there were samples in liquid that there were measurements, but it had to be sealed very well. Yes. In order to not so. Your question is completely right. So it is very difficult to have a real biological sample without an additional like fabrication and manipulation of the sample.
And looking at going away from biological samples, rather to go for, I don't know, a topological examination of superconductors, for example. Yeah. Could you then also, I think, do some with your energy comp, do a very innovative analysis which we have not yet thought of, I guess. Yeah, is this right? So definitely we need to connect. So.
Okay. We'll further talk.
Connection is all what we are looking for here, for connections and collaborations. And I think on top of what you were just discussing, having some experiments in live organisms, perhaps this technology with ultra fast EM, where you don't need to have it frozen, could also use for maybe protein dynamics and other type of drug discovery mechanisms. Would that be possible, Yaniv?
Yeah, so I believe so. It has a specific modifications. So for each sample, we can change, for example, the laser pulse that we use, the length, the duration. We can have many electrons that pass through the sample, or a single electron that passes through the sample. So I think that with the correct modifications, it could be possible to also look at different cells or proteins, again with what was talked about before with encapsulating the sample. Yeah, but it's a very good idea, and I hope we could have something in our experimental setup.
Great. We have some questions in the room. So first, Ulrich from Thais. What do you have on your minds?
Oh, it's. I also just want to understand it better. Like, if I get it right, the high temporal resolution is directly derived from the femtosecond laser. Now, what is the specific advantage to do the detection via the electron microscope? Wouldn't it be easier to just go with a light detector or a street camera or something like that? So, what specific signals do you get from the electron probe?
Yeah, so it's a great question. If you want a light collection, so definitely a street camera is much better if you want a very fast light collection. However, if you want to see in the near field what's happening inside the sample itself, and you want also high spatial resolution, then you need an electron microscope. So the electron can give you, it penetrates through the sample and can give you spatial resolution that you might not have in a street camera, where you collect light from the whole sample. You want to look at excitation at a specific point in space. And for this, you need an electron microscope.
Okay, now we also have a question from Oliver, our epic good friend Oliver from Bauer Innovations. Oliver, what is your comment? What questions do you have to Yaniv?
Hi, can you hear me?
Yes,
Yes.
very well.
Yeah, thanks for the great presentation. I'm just wondering if you can benefit from shorter pulses to get a higher time resolution, or how is it depending on the pulse duration of the laser?
Yes, shorter pulse can give us higher resolution, and it comes in two ways. One of them is that the width of the electron. So you see how I draw here the electron as a pulse. It also relates from the exciting laser. So our time resolution depends on how small our electron is in time and space. So with a short laser, we can have a very narrow in time electron. So that could give us a very high temporal resolution. And also from the other side, where we want to look at whatever we excite with the laser inside the sample. So for example, here you see this wave packet that I draw here. It has also some width in space. If we had a shorter laser, probably this width would be smaller. And then we could see finer features. So higher, like an attosecond laser could give another several advantages.
Okay, thanks.
Yeah, just to be clear, Jan, you're talking to the CEO of Valo Innovations, and this is the company, one of the companies has the record on shortest pulses lasers. They can make lasers on 50 femtoseconds or less pulse width. So we are talking about the application for microscopy is clear, and there is clear room for cooperation. Oliver, how can we make that happen? How can we get an experiment going with these people in Technion that they really would use your laser?
I think we could get in touch in the next days or so, and then we can discuss what we can do.
The question that is coming now is interesting for Yaniv, but also interesting for Oliver. Michele, for pioNIRS, thank you so much for being with us. What's on your mind?
I was just wondering what kind. So thank you for your interesting talk, Yaniv. And I was wondering what kind of light did you shine on the 2D material that you imaged?
Yeah, so the light that we shined here, yeah, it's a good question, because we shined in the infrared. In these materials, the modes that can propagate inside the sample are around the 6 or 7 microns. This is the wavelength of light in vacuum. So this was an advantage which looks somewhat technical, but this was the first experiment in the world with this excitation frequency. Most of the works until now were in visible range or near IR, and this was the first experiment, although there is also another group in the world that has the capability, and I expect them to have also some work that would be published soon. So This is in the infrared. So it's a good question because it is indeed unique.
Michele, wait, you're going to have a presentation later, but still, you're one of our key companies in time-gated spectroscopy. How do you think you could help the research of Technion at this moment? They are leading this field.
Well, I'm here. I mean, I'm loving listening to all those developments, and I don't know if pioNIRS can be of any help. I mean, we are working with pulse laser light, but we work in a time in the time range of picoseconds. So and we inject those pulses inside human tissue, for example, human brain. And here I was just interested in what kind of light was shown on this sample, because I mean, we also create laser light and we reconstruct the laser pulses. And so this is why
we're going to come back to you at the end. You're our final speaker all the way from Milano, but if you are all ready, we are warm up. It's 1 hour of the meeting. Now we go and we take serious presentation. We go to IBM. Thank you so much for accepting the role of being the speaker from IBM at a medical meeting on quantum technologies, Robert Loredo, a very well-known speaker from IBM. The floor and the attention of everyone goes to you.
Perfect, great. Thank you very much, Jose. Hello, everyone. And I know I have 6 minutes. I have my timer running. I'm ready to go here. So I'll just kick everything off here. My name is Robert Loredo. I'm the Quantum Technical Ambassador Lead here for the IBM Quantum. Basically, my overview here is going to be what we're kind of doing here with IBM Quantum in the areas of life science and healthcare. So just to sort of get started, you know, quantum computation, looking at it from a computational perspective now, it's in various different areas, both in chemical and finance. You know, it's just basically computing, right? So anything that you do is computational, and the information, the data that your systems are collecting go somewhere to get computed, or they're stored and then they're compressed, or they generate models from these, et cetera, et cetera. So there's a lot of work that's being done from a lot of this quantum, from a computational perspective, both classically and from quantum systems. So just to give you a little overview as to where we are so far. And the bottom here, this is sort of our roadmap and hardware and software roadmap. So just to kind of give you some where we're heading, where things are now, to get you kind of up to speed here. So you can see here now we're in 2021. Down at the bottom, we have various different quantum hardware and systems that we have going. We're currently now at a 65 qubit Hummingbird. We're going to come up later this year with one for 127 qubits, and then of course the next year 433 up to 1,000, over 1,000 qubit devices. Now with these particular perspective in 2023, we expect a big change here. This is sort of a very strong inflection point, mainly because the technology that we're putting into these processors are going to be now integrated. So which means that now rather than having a single qubit system, or devices and processors, you can stack these, as you can see here, where you can get from 1,000 up to multiple thousands into the millions. So because of this, then we have to also, on top of that, not just build the hardware, but build this sort of software that can run through these. So you can see on the left here, various different developer types. Kernel developers are those that are actually down at the circuit level. They're creating these runtimes, circuits, dynamic circuits, in order to be able to create these sort of circuit libraries that can be used by algorithm developers. And that's at the next level there. Algorithm developers are just that. They're looking at how to be able to take advantage of these circuits, this quantum phenomenon we have, and be able to generate sort of algorithms for different types of domains. You can see the different applications here we have for natural sciences, optimization, machine learning, chemistry, etc. And above that, of course, when we have the model developers, and these are the ones that use the algorithms. So these are the researchers. These are folks like yourselves, or anyone, you know, a chemical engineer, a photonic specialist, or maybe in this case, somebody in the life sciences who wants to be able to just get their research and leverage quantum. They want to, whether it's coming from a quantum computer system directly, an algorithm that's already been developed. So you can see that's where we're sort of headed in this particular way, in this form. So from that, you kind of figure, well, what kind of applications can we apply to quantum computing? So there's basically these three sort of buckets that we have where we look at which are sort of ideal quantum applications. So in the beginning here on the left, you can see is quantum simulations. So these are things that simulate physics and nature, things in chemistry, material discovery, material science, these sort of things, because they kind of have the very common components and foundations among them. Next up here is linear systems. So being able to do things like linear analysis, network analysis, differential equations. We're using it in various different areas, for example, in chemistry and in finance, right? Option pricing and heat transfer, and machine learning for things like classification. And finally here on the quantum walk, so this is graph properties, flows, electrical circuits, searching, collision finding, these sort of things. So these are all different types of methods or application types that we look into. So now, based on that, now focusing now into life sciences and healthcare. So with quantum, we're obviously looking to be able to use these particular properties, use the different types of applications and circuits to be able to come up with computational systems which can enhance a lot of the current work that's being done classically. So here, for example, customizing interventions and treatments, we can enhance a risk assessment. There's a lot of risk analysis because you can do a lot of simulations using Monte Carlo simulations, maximum likelihoods, predict drug responses at the cellular level by using a lot of the physical components on there, and of course, proactively provide personalized interventions here too, because if you're at the cellular level, you can definitely look at particular pieces. And then digging in a little deeper on that in the life science and protein folding and predictions, we're looking at basically understanding the structure and functions that are available in different proteins. We actually have a demo on one of our sites, which I'll be sending along with my PDF here with this stuff, so you can review. And it has a good protein folding demo, some papers that we've worked on looking at that, and as far as being able to predict or being able to analyze different proteins and their structures. So being able to enhance it with quantum, we could discover these new biological therapies that we go in there. And of course, just extending from there, being able to link genomes and then their outcome to sort of drive this precision medicine. We're looking at de novo structural predictions, machine learning models, which we can leverage to be able to interpret a lot of this data. And of course, coming up with possibly even digital twin models for most of this. And finally, here in healthcare, be able to accelerate diagnostics. So some of the work that, for example, image processing, which we saw earlier from Yaniv, we collect a lot of this data that is quantum based in a sense, right? It's a lot of sensor, a lot of information, but yet we get it and then we put it into a stored on a classical system. So we have to now convert from quantum to classical, and then to put it in a quantum machine, you have to now again convert it from quantum or classic to quantum data. So looking at being able to maybe monitor those directly. People have fiber optics in their homes now, so being able to kind of link those together is something that might be interesting to look into. But this basically does provide some accelerated diagnostics. And finally, of course, looking at the quantum network, and this is a question I heard here asked from Elena. With this group now, how can we collaborate? So we have a quantum network that we do it in order to be able to do that. And this covers a lot of things. One, obviously, is educate and train people that come into quantum or interested in quantum. We have a great program that's open source, as well as those that are directly with a quantum network. We also work with research to be able to accelerate a lot of this research and development that we do. And of course, develop applications. So we will be able to put these hardware, software, life science components together. Here's just a snapshot of about 140 members. I've highlighted the ones in yellow that are in the area of healthcare and life sciences. So we have quite a bit. And of course, there's MirrorX down here who does quantum sensing work as well. So we have a lot of different formats. And of course, whether you're a startup, you join as a hub, a member, there's various different ways you can interact. And with that, I will stop here and open for any questions.
Robert, thank you so very much for this quick dive into quantum computing and all the vast amount of applications and capabilities that quantum computing brings into healthcare world. So you did tackle just a little bit, but I would like to go a little bit deeper into understanding so how photonics community can also contribute to it. So what challenge do you have from perspective of all these capabilities and all these applications of quantum computing towards the people who are working with photonics technologies that do imaging, sensing, and solving other problems?
Sure. So we, from a computational perspective, obviously input comes from everywhere, classical and quantum. So part of the work or some of the work that's actually being worked on now research-wise, from an image perspective, is image processing, right? You capture a lot of information, but now how do you analyze it? I mentioned a little component here on diagnostics. The volume of, if you look at a lab that looks at and analyzes images to determine if it's looking for cancer, looking for some kind of patterns in there, that requires a lot of work. And these centers usually get a lot from a lot of different clinics and sources to be able to send them their images. They want them to analyze it and give back a report or something, or some discoveries. So that's a lot of volume of information just needing to process. So speeding that up is one area that can really offer some benefits right away. Of course, on the hardware side, if you have a quantum sensing system or a system that gathers information that's quantum in nature, Like I mentioned earlier, there's that process of quantum, classic, and then quantum. So there's 2 points where you're sort of converting data and then converting it back to quantum. So another one could be maybe when to start from a quantum system, direct it, send that information directly, couple it to a quantum computer, with fiber optic cables or what have you, right? So just being able to have that stronger connectivity, so you skip that step of going classical and back if you don't need it, and if it's possible to do that, that also could speed up a lot of the processing information, et cetera. So there's lots of different ways to be able to look at that.
Okay, that is quite interesting. So we are looking into having a way to generate quantum data and transfer it directly in quantum. Jose, what do you think? Do we have anyone who can help us on this?
You know what? IBM has been one of the stars of the quantum ecosystem. You've been one of the first ones to give qubits to universities, and actually that has created a lot of success on this industry. When it comes to medical, and I was paying a lot of attention to your presentation. When it comes to medical, there are obviously societal problems to solve, but what companies and what universities can get from IBM on the medical R&D is not that clear. Is that something that we can actually maybe work out an example? And I have, for example, here at University of Leeds in the quantum sensing making a big effort. I have Technion. Let's try to make an exercise. Let's see how they could help you.
So I'm using IBM Q in a weekly basis. So I am definitely using mainly for teaching quantum technologies. So for teaching quantum technologies, this IBM Q is just perfect for us. But I think also for research, there is a lot of things that you can use IBM Q for. IBM Q is the platform of the quantum experience, the quantum, the quantum computer. So for us, it's very good for in research. From one point for teaching, but also quantum simulators and also, well, I don't know exactly how to do the machine learning, but I'm sure that my friends from the computer science division know better. And I know that IBM helps a lot at this point.
Thank you very much, Yaniv. And it's great to have gone up IBM thousands of users in the R&D side here in the room, using it for teaching. I want to go to my friend Professor Joseph. Jing, you have seen the presentation from IBM. They provide qubits for R&D. You have done lots of research on quantum sensors and actually sensors, high efficient sensors for biodiagnostics. Do you have an idea or a mindset that could benefit by a collaboration with IBM?
Yes, definitely. Yeah, I think need to know more about it. I think what Yaniv said was very interesting. So starting with the support for teaching and then knowing better what it can do will really help in progressing further.
Robert, in the opening presentation of our quantum meetings, we actually had this announcement for the extra qubits being given to the industry, given for new research projects, not only for research, but there's also large amount for industrial cooperations. It is so great to have IBM in the room. Whenever you spot no other potential cooperations for the medical industry, and we want to have more meetings on quantum computing, which I know you're going to be there as well. But whenever you spot the cooperations, please let us know, raise your hand, and we'll make it happen. It is great to have you in the room, Robert. I know you are very busy, but I also know that you are a keynote in many of the quantum technology presentations. I always meet you in the lobby, so I was so happy to have you today in my room. Thank you so much.
Pleasure to be here. Thank you.
And know what we're going to do now? We go to one of the companies who you have seen in every lab. We go to one of the success stories of the quantum European industry. We go to the world record on quantum detection, the world record on single photon detection, and we go to Delft, the Netherlands. All that in one presentation. We go to Sander from Single Quantum, a good friend of mine, and also a proud member of the quantum photonic industry. The world is watching you today, Sander. Thank you very much.
Thank you so much.
The world is watching you and hearing you now. Yes.
And thank you for inviting me for this very nice and interesting meeting.
Thank you for holding the world record. It's not every day that we get this in our room.
So my name is Sander Dorenbos. I'm the CEO of Single Quantum. And our mission is to develop the world's fastest and most sensitive light sensors, limited only by the laws of physics. And this basically means, more concretely, the best detector specification, as in high efficiency, so detecting all the photons, low noise, short dead time to have a high count rate, and a high time resolution that probably most of you know would give a very high spatial resolution. And the way we do that is you're using superconducting nanowire single photon detectors. As the name says, this detector consists of a very small nanowire, of which you can see a SEM picture on the left. And this wire is brought in the superconducting state. And once a single photon is absorbed, then a small part of the wire is converted to a resistive state. And this can be measured by sending a current and basically measuring the voltage. So this chip is packaged and fiber coupled. You can see in the middle, and it's placed in a cryostat, to closed cycle cryostat, to reach the right temperatures. And these days you can put up to 24 of these detectors in one cryostat. And as Jose mentioned, these days we claim to have the world's highest detection efficiency, as you can see in this quite technical graph. On the bottom, there's the bias current that you run through the device. And when you increase the bias current, you can see on the right the dark count rate, or the noise, the black points that ramps up, but still at high currents, the dark count rate is very low. And on the left, there's the system detection efficiency for more or less different wavelengths, but around the telecom. And as you can see, the higher the bias current, it can these days almost go to 100% system detection efficiency. And well, the main advantages of these detectors are that this high efficiency can be combined with a very high time resolution and also high count rate. As you can see here on the left, you can see the time resolution that is now around 10 picoseconds, and on the right, you can see the detector count rate. The detection efficiency as a function of the detector count rate, and even for the highest pulse rate of our laser, it reaches almost 90% efficiency. So what are they used for? Yeah, of course, nowadays they're mostly used for quantum communication, but we also want them to be used in imaging. And the basic idea is that when you want to image into a biological sample, the optical access is limited by scattering. So if you want to image deeper, you need to go more to the infrared. And of course, it's difficult because then you are limited by the sensitivity of the detectors. Our detectors can solve this. So here you can see a very conventional example where a mouse heart is imaged. I think this is a few millimeter dip, and compared to a photomultiplier, our detectors perform much better. But of course, this is not quantum. So since we can detect single photons, it can also be used for some futuristic technologies, quantum microscopy, where you use single photons or correlated photons, or in the best case, entangled photons to make images of objects. And this is an idea that we are developing now with European funding called FastGhost, where entangled photons are used, with photons in the mid-infrared will be interacting with the sample, and then detected with our detectors, and then the entangled twin that is invisible will be detected by array. So basically, ghost imaging. And with this we believe we can image deeper and with higher resolution in the sample. So I think we also I was also asked to name some challenge. So yeah, what is challenging in this still is because our detectors are fiber coupled, we are still looking for ways how to fiber couple this near infrared or mid infrared wavelength.
Thank you very much for this quite inspiring presentation about microscopy and actually looking beyond the object with a fast ghost. That's really amazing and fascinating. And yes, it's an example of the mouse. It's like, wow, I wish I had the detector like that when I was doing my research. And it looks quite obvious to me that this single photon detectors have a lot to offer to the research and to the development of new drugs or new imaging techniques within healthcare. So as you already know, the epic style, epic attitude. So we did hear quite well what you have to offer, but then let's see what actually the supply chain and people who are present in the room with us can help you with more.
So, but I have a question first for Sander, because I know this company already for quite some time. I don't want to say, but I think it's since 2014 the first time I met your colleagues. We've seen each other at Photonics West and other conferences. I am quite impressed and amazed, and I would like to congratulate you for what you are achieving. But what's next? What's next? You got the world record already. What's next? Is miniaturization? Is room temperature product lines? What is the next thing that is coming from you?
Now, for me, the next thing is, well, now we have a single pixel, and the next big thing will be a camera.
A camera, a single photon camera operating at this resolution. Oh wow! Yes. And what kind of partners could I introduce you to make that faster, earlier, cheaper? What do you think? Dream away. It's the Santa Claus Christmas Day. Tell me.
Yeah, that's a good question. I guess some kind of electronic integration is still very challenging because you get a lot of data.
I have a lot of that here. I'm going to introduce you to a few of my friends. I'm going to go first to CPI, to Kath Hossein. Kath, thank you so much for being with us this beautiful afternoon. It is 4:22 in the Netherlands, which makes 3:22 in where you are. So Kath, did you see the presentation from Single Quantum? You came to this meeting to talk about quantum microscopy. What's on your mind? What room for cooperation can we actually exploit? We can't hear you. And I really want to.
Can you hear me now?
Yes, loud and clear.
Okay. Well, in terms of quantum detection, I don't think we have currently any specific requirements, but it's all going to feed into what future healthcare is going to look like, because everybody wants increased sensitivity, but at the same time, they want it cheap, small, and mobile to do continuous monitoring. So that's kind of the main focus of the vast majority of our work at the moment. But I can also see additional research lines coming through of interest that actually require very high sensitivity that you're not going to be able to do in a small mobile piece of equipment. And for that, the specialism of being able to do something like single photon detection for very, very sensitive maybe even fluorescence assays where you're looking for tiny samples that might give us an edge in terms of diagnostics. So novel diagnostic systems could certainly be a future application that I can see for this level of sensitivity, and that's actually quite exciting.
Kath, what is in broad lines, general lines, what is CPI doing in the quantum tech?
There's nothing specific that we are doing. We are very much looking for quantum tech that can support. I mean, we have a wide range of business areas, from printable electronics to formulations to vaccines. And it's finding ways of optimizing our processes and technologies by supporting them with, for example, quantum computing, which could give us an edge in terms of non-destructive testing, machine learning. We know that there's a lot of evidence that AI is actually more accurate when it comes down to sort of image analysis. As soon as you put the human in the loop, you get individual variations, and it's little things like that that can potentially push what we are trying to do into more efficient overall systems. So there's a lot.
Thank you very much, Kath, and I can't wait to come and visit. I had to start traveling again. You know, if there wouldn't have been this horrible pandemic, we would have this year the World Quantum Photonics Summit of EPIC hosted in one of the most beautiful areas of this beautiful continent called Europe. We will have it in Glasgow. Kevin McIver, thank you very much for being with us today. You just heard the presentation from the world record of single photons. I know you are really active on many of these technologies. I don't know if you have a single quantum device, but what kind of room for cooperation have you spotted already? What kind of things can we do together here?
Thanks, Jose, and I'm delighted to be able to speak to Sander and see him in person, virtually, of course. We have spoken via one of our colleagues, Robert Hadfield, and I understand that we are close to collaborating on a PhD studentship, so that's very exciting news. So the collaboration is already there. Given your presentation, Sander, I think there are a lot of synergies in what we're doing at Quantic, in particular in the University of Glasgow, where we have the 3D quantum imaging in place. We're also looking at microscopy with quantum illumination, where we're replacing the classical illumination where photons arrive one by one with quantum illumination, where the photons are arriving in pairs, these photon pairs acting as both probe and reference. So there could potentially be somewhere there that we could speak about if that's appealing to you, Sander. So certainly we can chat offline from here. Jose, great meeting as usual, and it's been fascinating listening to all the presentations.
But let's all get vaccinated and have the real meeting in Glasgow, because I really want to meet all of you again, but real in person. Let's all of you get vaccinated as soon as possible. I would like to now continue with this, but I would like to understand a little bit about the commercialization, Sander. So at the end of the day, many of your customers today are in the R&D side. I see a lot of your equipment in the labs that I visit. What is the next step to have more industrial customers? What do you think we need to do from the EPIC side to outreach the industry, which is the reason why we are doing quantum meetings?
Yeah. Basically, what we're waiting for, of course, is for quantum communication itself to be industrialized. I think we have
quantum communication industrialized. If that is the case, I actually have a very important person in the room. I have Nick from Quantum Valley Ideas Lab in Canada. Nick, how are you doing today?
Great, Jose. How are you?
I'm doing great. And when I see you all the way from Canada, I get super happy today. It is 4:28 in the afternoon here in the Netherlands. What time is it for you?
It's 10:28 here, just outside Toronto.
So you just heard that Sander wants to, he says, if there is a commercialization, it's because quantum communication, QKD, has to be closer to the market. What is your vision on this?
Yeah, I think So this issue, right? I think it comes through in a bunch of the conversations today, right? Is the quantum, end user market versus the commercial end user market, and when do we transition from that sort of experimental market where people are leveraging quantum for a set of use cases versus something that has a general broader market. And I think some of the discussions earlier were similar, right? In terms of when will this replace an as-is technology in a broader market, right? And I think from our perspective, we're pursuing some projects in different areas, but also looking for opportunities to commercialize in some of those other areas, right? And I think there's a lot of activity here in Waterloo on communications and QKD work. I still think there's some startups and some activity, but from what I've seen, right, there's still sort of this evolution of timing to get towards more of a mass enterprise market kind of opportunity.
Thank you very much, Nick, for being with us. Sander, I got super excited when you told us about the single quantum camera. What kind of applications, what kind of problems do you think this camera is going to solve?
Yeah, basically in the medical, for example, in the biomedical field, where you can image deeper at higher resolution.
Sander, you know what? I love when nice things happen to nice people, hardworking people. You are a quantum hard worker person that managed to actually put something very innovative in the market and get the recognition from the entire industry. It is an honor to have you in the room today. Thank you so much.
Thank you.
And we go now to a new one. So we have a very well-established single quantum company, and we go to one thing that is making us all excited, all the way from Italy, all the way from Milano, a new startup. And I love this. pioNIRS. Michele Lacerenza, thank you so much for being with us. Grazie mille for being with us from Milano. The floor and the attention of everyone goes to you, goes to pioNIRS.
Hi, Jose, and hi, everyone. So
this is our final speaker today. You know how fast these meetings go, right, Michele?
Yeah.
So far, have you spotted some room for cooperation? Some people that you want to have a chat afterwards?
Yes. I think there is plenty of opportunity on the table. And
let me remind you and everyone that when your presentation finishes, then we are going to have a short summary of 1 minute, maybe 30 seconds, and then we are all going to go to this new thing, which is we're going to have a link in the chat, which we are all going to click. Then we close Zoom and we appear in a new environment in which we all can actually talk to each other and find ways for cooperation. Let's try it. I tried it in a different event before and I loved it. Let's try this. So after this presentation and a quick summary, you're going to get this link. Just click on it, close Zoom, and then we have our closing chat. I wish we could all have a beer at the same time. But now, pioNIRS. This is a big one. Michele, thank you so much for being with us. The floor is yours.
So thank you, Jose. And so here we are. My name is Michele Lacerenza, and I am co-founder and CTO of pioNIRS. I'm going to talk to you about the technology that it's in the heart of our company and some of our products, together with some of the application that we have and a vision to the future. So as everyone here knows, light interaction with matter, especially biological tissue, can give us paramount information about what we are looking at. And especially light can penetrate in our body, in fingers. And for example, it can unveil hidden information in biological tissue, as we see in those images. We can have information about intensity of light, but there is a full other understanding of what light can tell us in other kind of domains. For example, what we work on at pioNIRS is the domain of time. We generate very short laser light pulses, very short in time, talking about picosecond, not femtoseconds, as we were talking about earlier. But still, those kind of light pulses are injected inside the human tissue, for example, the brain, and then recollected. By analyzing the difference of the shape in time of those pulses, we are able to retrieve important feature of the tissue that we are looking at, chemical composition and physical composition. And sometimes, depending on the wavelengths that we are using, we can retrieve the concentration of different chromophores that in our case here, we have the concentration of oxygenated hemoglobin, deoxygenated hemoglobin, and tissue saturation. But how do we do that? So when photons are injected in the tissues, they travel and get back scattered out of the surface again. What we do is using optical fibers to collect those photons and bring them to the active area of a quantum detector. The quantum detector we developed are called silicon photomultipliers. So they are based on these technologies. And so the single photon information is transferred to a time-correlated single photon counting board that we custom developed at pioNIRS. And we sort out these photons in order to reconstruct the laser pulse in time. The features are very interesting. We're talking about 100 gigahertz equivalent bandwidth and a count rate that reaches 1 million counts per second with digital-like robustness. So we work in it, as I said, in a domain of time. And why? Because time encodes depth, time encodes penetration depth in biological tissue. Early photons brings us information about the superficial layers that we are looking at, while late photons can tell us a lot about deeper layers. For example, reaching 3 centimeters depth on cerebral cortex in humans. The unmet clinical need that we are tackling, the first one we are talking about, is the concentration of oxygen in tissue, in deep tissue. And time domain NIRS, near infrared spectroscopy, is pioNIRS technique that can tackle this unmet clinical need. How does it do that? Being cost competitive compared to state-of-the-art devices such as fMRI and PET technologies. Being non-invasive and enabling continuous monitoring, even in those conditions in which, for example, little kids cannot stay still and cannot acquire, and so it's not possible to acquire high-resolution images. The device we developed is called NIRS box and is based on pulsed laser diodes that we couple the light into optical fibers. We send the light on the tissue and we collect them with other fibers, sending all the photons to the single photon detector. Compared to the other instruments in the market, coming from the university and with 20 years of background on these technologies and technique, we have great stability, accuracy, and reproducibility that makes our device suitable for the clinical environment. So let's go through some of the features of the device. The first one is precision. Being precise in acquiring such information make us able to retrieve even the smallest changes on functional and monitor functional cerebral activation. For example, here we see the right hand is moving and the left hemisphere of the brain is activating. And what we see is an increase in the concentration of oxygenated hemoglobin and a decrease in deoxy, while we see no activation when the no signal when the activation is on the other side of the brain where we are not looking at. The second feature is being safe. So our device is very safe. It has got very low power laser sources thanks to our quantum detectors. And being safe means to be suitable to clinical environments such as neonatals intensive care units. So those are this is one of the projects where time domain NIRS is employed. And in this analysis, little like newborns, babies, the health of the brain of newborn babies was monitored during red blood cell transfusions. The third feature is accuracy. So being so much accurate make us able to enter the clinical environments related to, for example, breast cancer diagnosis. So here here is an example of how small those devices can be, and how effective can be a multimodal integration of different technologies. Here we are using time domain NIRS at different wavelengths and ultrasounds to have better and to boost the diagnosis of breast cancers. This product is now through clinical validation. So a glimpse to the future. We managed in a couple of years to go from very bulky devices, such as big fridges, based on time domain NIRS technologies, to a device that we have right now in the market that is as big as a small book that can monitor brain activity while people are moving around. In the future, we are looking for shrinking the dimension even more and make this technology accessible to the world, everyone, everywhere. That's our team, two executive, me and Mauro, and then our great scientific advisory board, together with our amazing industrial partner, that is Microphoton Devices. So I thank all of you for your attention, and I'm open to any question.
Thank you so very much for this great presentation, and really fantastic how you can use the technologies to just go through the brain, through the whole scalp. So I just want to understand how deep it can go. So does it measure indeed through, let's say, if we are talking about brain imaging, through the whole organ, or there is still some limitation there?
So thank you for your comments and your question. As Sander said before, the main limitation is related to the scattering. That is an intrinsic feature of our body and of diffusive media. So the maximum depth we can reach right now is at around 3.5 centimeter. And this depends a lot on what source detection separation you select and how high is your signal to noise ratio. So depending on those technical features, theoretically we can go even deeper. But right now, what we are stuck to is 3 centimeter penetration depth, approximately. And what time domain gives as an amazing feature is being able to select the penetration depth. So being able to disentangle the information from upper and lower layers. And most of all, disentangling absorption and scattering coefficients. That this is one of the greatest problems in clinics right now.
Okay, thank you very much. I would like now to get back to our medical device manufacturers and also and to see if, what do you think about it? For instance, Sergey from GE, you heard now about all these different technologies, and some of them already they do have end application and they're available there. So you came here for this meeting also to learn about what quantum technologies are there and what they can bring. So what feeling now you're getting at the end of the meeting?
Yeah, so some of the technologies, specifically last presentation, it's obviously it's very, very close already to application. I mean, to clinical application. Specifically, I found, okay, so oxygen level measurements in the brain, it's really, they already have prototype devices and it's close to the commercialization if it's not commercialized yet. And the second is for breast cancer, combining device with ultrasound. I believe this is very, very interesting approach. I mean, because you have two different signals, which is overlay, and it gives you extra information, which is always useful. We try to combine ultrasound with CT, ultrasound with MRI. Now you have combination ultrasound optical. I think this is very good potential specific with that one application. Obviously, quantum computing is totally different story. So it's computing. It's just you should consider computing. I'm more interested about sensor levels. Yeah, yes, for how you can apply technology for sensors. Fast detection with 95% efficiency for a photodetector with superconductive wire. This is Dorenbos presentation. It's very nice. It's a nice tool which I would have in my lab just for characterization of some light sources. I don't see application-wise, but it's really, I would love to just have it like, okay, it's a little bit tricky to use. You have to fiber it. So it's interesting just join these forums like that and see where is all direction is going on, all the development is going on. Obviously, some from the science point of view, where is limits, where is quantum, all this entanglement and all this stuff, it's really plug in inside, or it's where it's only just name, it's quantum, everything quantum. So I like this forum. We have a couple site connections already through the chat room. So I will follow this couple with Yaniv and with Mathieu, I believe. So, and interesting. Thank you.
It is fantastic to hear that there are already cooperations being created even before the meeting is being done. That's what we're all about here today. And I also want to go with a similar question to Thomas from Merck. So what do you think about this time-gated near-infrared technology as that Michele was presenting, and in general about all the technologies that were here discussed today?
Yeah, so first, the second part of the question, I think it's fundamentally interesting to see that there is so much interesting technology out that is hard to sell to the healthcare industry because it's not obvious what this can do. Yeah, and I think if there would be more meetings like this with more participants like myself, this would help out. But I mean, it's a start, Jose. Yeah, you never know. For the specific question to Michele, I really am fascinated by the approach to do this this kind of spectroscopy. And the concrete question I have is one of the fundamental paradigm and problems is the blood-brain barrier. Yeah, because nobody understands how it works, and it's crucial for drug transport into the brain. And for the nervous system in general. Would it be able that this kind of technology can be used for better detecting how it works? Because nobody has an idea how it works. There definitely are quantum effects involved, but we don't know if it's tunneling or how the brain barrier actually really works. Did you already investigate if this is possible at all?
I have a question upon yours one. What do you mean about brain barrier?
There is. I mean, if you swallow a drug, we have a blood-brain barrier, which is a kind of smart membrane, which lets in glucose but not toxic waste. And if you want to treat your central nervous system with specific drugs, you need to make the drug penetrate the blood-brain barrier. And if this doesn't work, you can't just. I mean, the nervous system protects itself. And there are lots of studies there are millions of studies published on hypothesis how the blood-brain barrier could work, but it's really one of the mystics. So
yes, that's interesting. I mean, super interesting to know about it. I didn't know about that, and what I can think about when you're talking to me about this kind of problem is that we could have two different approaches. The first one is being able to actually detect the chromophore that wants to break this barrier and see if this element that has been injected actually can reach the tissue and the microvascular network. And the second approach is whether this kind of barrier breaking triggers an hemodynamic changes. So if that is the case, then these technologies, I can say it right now that these technologies can have a good potential in this kind of.
I think there's definitely something we need to follow up offline. But my understanding of how it works is that there are lots of pumps and ion channels, and even though it penetrates something, there is another channel that pumps it out again. And then the question is, of course, of spatial and temporal resolution. Picoseconds sounds good for temporal resolution. For spatial resolution, I think we have also other guys who can do spatial resolution very good. So I think there might be interesting opportunity for joint research if ever somebody is willing to fund this.
Yes. Thomas, but I think you're basically targeting kinetic studies, yes? So you basically So an IRS will not give you any special resolutions, and time and resolution picosecond will not help you. I don't see how. But what you really looking for using this technology, you're looking for how fast you wash out your biomarker, your drugs, and if it's penetrate your barrier, it should stay longer. So so what you really do, you do just using this technology. It's fine. You just adjust your drugs, which is has response signals on the IR. With wavelengths, and after that you just kinetic. You just do it comparison if it's okay. You know, some drugs which is penetrate barrier, it's have one kinetic study of kinetic behavior, and the other does not penetrate. It's wash out instantly. It's follow the blood, and you can do it's like two different regions or whatever, and just compare kinetics.
Yes, and the question was, would we have a method to understand how the blood-brain barrier works?
It's a question to you. This is tool to just help you to give you kinetic response, and after that your drug developer,
and then the miracle happens. Yes, unfortunately. Okay.
Yeah, but it's not physics question. Yes.
Yeah, it's not a physics question. That's right. I mean, eventually it is, but then it's kind of different kind of physics. Yeah.
But very likely photonics can help all of it, whether it's with pioNIRS or whether it's also with the help of quantum computing. I think when we come together, there is no question we cannot answer. What do you think, Jose?
Yeah, I'm so happy for Michele, because not every day you get, GE and Merck giving you advices, and it's so great what we are doing here, bringing the companies, the industry to the quantum technologies, trying to do a market pool of quantum. I know it's difficult, but that's our dream, and we're going little by little. I want to go to Cambridge Consultants, and I want to show him this slide, because for me it is clear that this is one of the follow-ups of this meeting, one obvious follow-up. So they are looking at how they can go into the monitoring of the brain for the future in a miniaturized way. There is a lot of regulations to go through this. There is a lot of R&D work to go through this. There is nobody better in the world than Cambridge Consultants to support this. How could you help a company like pioNIRS?
Yeah, it's really exciting stuff. I think we can help by looking to see both the feasibility of the miniaturized processes, but also we have teams which work on the integrated electronics and photonics and all of the miniaturization which is associated with that. Clearly, there's also the medical, the regulatory procedures, and we're very familiar with that. We have decades of experience with making sure that these things can have the highest chance of passing clinical studies, and I think it's really exciting stuff.
For those of you who don't know Cambridge Consultants, because in the medical industry they are very well known, this is not a consultancy company. This is 1,000 employees worldwide who actually have manufactured many of the current clinical equipment, making sure they go from TRL to 5 to TRL 7, go through all clinical trials and regulation process. This is a large company that we have in EPIC. You have supported us a lot in the medical industry. Thank you for also supporting us in the quantum medical industry. We are really at the beginning of something truly great. Yaniv, Kurman, Technion, you have amazed many people. I'm getting a lot of comment private into how amazed they are with your presentation. You have talked to many companies today here. What is the immediate follow-up that you're going to do? What is the first thing you're going to say, okay, we need to talk to this guy and do something together?
Well, first of all, I'm a researcher. So the first, if there is an open question somewhere, then I'll try to maybe help and see, for example, to have a better photo detector in PET machines, or to see how each one of these people who talk today have different input and different suggestions. So I think all of the connections are very interesting, and the talks are very a lot of input for me. So it's very exciting.
You have a very interesting comment during the presentation from Sander from Single Quantum. You had a very interesting comment in the chat. It is the right moment to voice it out. What was on your mind?
I'm not sure which.
Single photo detector cooling is problematic.
Oh, okay. So yeah. So I recently heard a presentation I believe it was Xanadu, or one of the companies who are doing, startup companies who are doing optical quantum computing. So of course, in quantum computing, you can use different platforms, and it's currently unknown which one is the best, whether it's a superconducting qubit or photons. And if you want to do it with photons, then you need this. One option is to have a single photons, and for this you need a single photon detector. However, it is not clear still for the scientific community whether it's actually needed to have single photons for quantum computing, or you might have quantum computing with more than a single photon. And therefore you don't need a single photon photodetector. So just a last point for quantum communications, I do believe that single photon photodetectors are a must.
For many of the applications that we targeted today, we don't need single photon detectors in the high extreme case that the single quantum presented. Actually, SPADs are a great tool for that. I would like to go, I know he seems busy, but he's never busy enough. Nick from Quantum Valley Ideas. Nick, to what extent we can use SPADs in QKD networks, in quantum communication? Do we need these cryo cooling single quantum detectors that we have seen today?
So our focus so far has been more on RF sensing using Rydberg atoms. So we've been sort of trying to focus in on room temperature activities, right, in general, right? We think, back to this discussion around solving for, the quantum market or solving for the commercial market, right? I think there was a lot of discussion earlier on swap and those kinds of topics, right? So I think there is, the commercial market, I think, will push people more and more, like the end user commercial market will push people more and more to more practical room temperature portable solutions. Like that's kind of our direction now. There's obviously use cases there that require the superconducting nanowires, but I think in general, eventually if people are going to buy these things for commercial settings in a broad market use case, you got to push towards more compact, more room temperature capabilities, I think.
This is our third quantum meeting, and so far my big learning is that many of the problems that quantum technologies have can be solved with very mature photonic technologies. I want to finish this meeting with the detector and the laser side. I want to hear from Richard from Excelitas and then from Yari from Modulight about what resonated with them during the meeting, what kind of room for cooperation, and then I will tell you my learnings. Richard.
Well, again, like Sergey and others, and like I said before, it's still the learning, but there's plenty of room for detectors where it is single photon. I think the other comment that some of the quantum here is not single photons, so perhaps there's less of a quantum interest here for our detectors than some other quantum areas which really are single photon. We're mentioning cooling. Obviously, then you just get into that trade-off between the quality of detection, low noise, low or to zero dark count, and the things that you're going to get with superconducting ultra cold, against something which is practical and usable, especially if you want to scale to volume and make it small. So, you look at comms, a lot of that's still going to be around in-gas SPADs at 1550. They're not going to be as good, but they may be good enough, and they're going to be much easier to use. And that's going to be the big challenge for the nanowire guys. If they can get a room temperature superconductor, for example, then the problem, then it becomes a different challenge. So, there's a lot of interesting and different developments. It's far from mature, which makes it really exciting to see.
10 years time to market, 10 fantastic years to have fun. Yari from Modulight, thank you so much first for sponsoring the meeting, and second for making some of the best semiconductor lasers that I can find in the market today. Yari, what kind of cooperations, what resonated with you in these last 2 hours?
Yeah, hey, thank you, Jose, and thank you everyone. And well, as a company, Modulight, we consider ourselves like front runners for the photonics usage in the medical industry. And we are all the time looking how to harness these new technologies to provide more intelligent solutions and serve the medical industry better. And as a company, we have this ambitious mission. So we are trying to kill cancer. And this is a too large task for the one company, and innovations rarely happen within one company. So we need partners, and really looking forward engaging with the participating companies. And I must say that I'm really surprised to see that we actually might have 3 different roles within this community. So we might be a supplier. So we have this single laser diodes, and we can supply these complex and tight pitch laser arrays or subsystems. But then, in the other hand, we have our own devices, which we consider to be like innovation platforms. And those platforms, we have been developing our internal software and algorithms. And it would be really great to leverage the work what the other companies are already doing. And really on this Kill Cancer mission, it's something that we definitely need partners. So we can be a supplier, we want to be a customer, but we want to be a partner. So thank you, Jose, for
Thank you very much, Jerry, and thank you very much, everyone. These are my learnings. Qnami. Qnami is looking for partners for miniaturization and affordable volume production. The University of Leeds, they presented this amazing, Professor Josie, amazing non-invasive glucose monitoring. He's looking for partners using more accurate detectors. We have Technion, they had great job in ultra-fast electron microscope with spotting some possible cooperation in ultra-fast lasers, femtosecond lasers, single quantum. They have a world record on single photon detectors, but they were here with a very specific challenge. We want fiber coupling and suppress the black body radiation. And finally, the best, we say the best for the last, pioNIRS, very clearly. We want partners who do trials for oxygen level measurements in brain. That was really, truly fantastic. Now, we're all gonna get a link in the chat. All you have to do is click there and close the Zoom and just follow very quick instructions in the browser. It will be totally worth it. We are all gonna appear in a networking environment. This is all about making connections. This is what we want to do here. We want to make connections. I live in Northvik, next to the sea, and every day the sun is brighter, and every day I'm closer and closer to be vaccinated, and I start traveling again. I used to travel 200 days a year. It was amazing. I can't wait for this to continue happening again. If you want to get in touch with any of the participants today, just send me an email, jose.pozo@epix-aso.com. Everybody that was in the Zoom room is looking for cooperation. That's what the EPIC meetings are about. We love hosting these meetings in quantum technologies. We also organize many of them in the photonic industry because that's what we are all about. Here are the upcoming meetings. Write down the one on Monday, 15th of March. LiDAR applications. 180 companies already registered. Until the next time, on behalf of a fantastic team of medical experts, integrated photonic experts, laser experts, quantum experts, an innovation manager from Italy, Antonio Raspa, who's totally crazy, public relation managers, event managers, a marketing manager who knows everything about communication, Ana Paez, Carlos Lee, and myself, the geek. Thank you so much for making EPIC a quantum business. Until the next time, wash your hands, wear a mask, and take care of each other. See you soon. Bye-bye.